Hello, everyone. Welcome to another exciting edition of Hematology and Blood Transfusion Science Lecture Series. This is a YouTube channel where we discuss different topics in hematology and blood transfusion science. Thank you for being part of what we're doing. Thank you for subscribing to our channel. And if you have not yet subscribed, this is the right time to do that. Thank you very much. We will be starting a series of lecture on the topic hemostasis. And today we will be handling particularly the topic that says primary hemostasis. Primary hemostasis. My name is Dr. Ifeima Mary Ann Okafor. Welcome once again to my channel, Hematology and Blood Transfusion Science Lecture. Thank you very much. It is expected that at the end of this lecture, you should be able to explain the role of vasoconstriction in hemostasis. You should be able to describe the mechanism of platelet adhesion, platelet activation, and aggregation. And you should be able to identify key laboratory tests used in evaluating primary hemostasis. What is hemostasis? Hemostasis can simply be defined as a vital physiological process that prevents excessive blood loss after vascular injury while maintaining blood in a fluid state under normal circumstances. It is a physiological process that prevents excessive blood loss after vascular injury and is also has the function of maintaining blood in a fluid state under normal circumstances. Hemostasis actually depends on a precise balance between clot promoting and clot inhibiting mechanisms, or you can say a balance between procoagulant and anticoagulant mechanisms. I want us to distinguish properly between two processes, coagulation and hemostasis. Hemostasis simply refers to the overall process of stopping bleeding or hemorrhage, while blood coagulation specifically refers to the process of forming a blood clot, and it involves a complex cascade of enzymatic reactions that convert liquid blood to fibrin. Now, hemostasis is the broader process of stopping bleeding, while blood coagulation is a key component of hemostasis that involves clot formation. There are stages of hemostasis. For ease of understanding, I divided the stages into four. We have the vascular constriction, platelet plug formation. The platelet plug formation can also be said to be primary hemostasis. We also have coagulation cascade. The coagulation cascade can be called secondary hemostasis, and we have the fourth stage, which is fibrinolysis. Fibrinolysis can also be said to be tertiary hemostasis. So we have the first stage, the vasoconstriction or vascular constriction, the platelet plug formation, which is the same as primary hemostasis, the fibrin cord formation or coagulation cascade or secondary hemostasis, and we have the fibrinolysis, which is the tertiary hemostasis. So, four stages of hemostasis. Let us go straight to what we are going to be discussing today. Primary hemostasis. Primary hemostasis. And we will be starting with vasoconstriction. We will be starting with vasoconstriction. The first response to vessel injury is vascular constriction. When we sustain injury, the endothelium is disrupted and smooth muscle cells in the vessel wall contract. Now, this contraction is mediated by local nerve reflexes, by circulating mediators such as serotonin, thromboxane A2, and endothelium, released from the healthy endothelial cells surrounding the region of the injury. Now, the narrowing of the vessel lumen reduce blood flow to the site of the injury, limiting blood loss, and at the same time, buying time for the next step of hemostasis. When you have vascular injury, there is immediate narrowing of the injured vessel, which is mediated by endothelium, neural reflexes, serotonin, and thromboxane A2. Now, the essence of this narrowing of injured vessel is to limit blood flow to the site of the injury and thereby preventing blood loss. And all this is done before platelet activity begins. Now, we don't have any direct tests that are available for vascular constriction, but we have other many other techniques that we can use to access vascular reactivity, especially during research. Okay, but for diagnostic purposes, vascular constriction occurs as an immediate response to injury. We can observe the response or assess the response indirectly through the observation of initial bleeding time in certain circumstances. However, we have some laboratory tests that can be used to evaluate vasoconstriction, even if it's for research purposes. So we have number one, vasoconstriction assay, which is scale blotting assay. This is just a simple non-invasive method that is primarily used to evaluate the potency of topical corticosteroids. Now, the drug is applied on the skin, usually on the forearm, and the degree of vasoconstriction is visually or instrumentally measured by observing the color change on the skin. You can also apply the use of instruments like chromometer that can quantify the amount of color change. We have another technique that can be used to assess or evaluate the vasoconstriction, pressure myography. The pressure myography involves cannulating a blood vessel segment to measure its contractility and reactivity. A segment of blood vessel is carefully dissected from an animal or human tissue, mounted on a myograph, and its diameter is measured as vasoconstrictor agents like thromboxane A2, serotonin are added to the surrounding fluid. Now, this method provides precise data on vascular reactivity without the influence of other factors. We have also another technique that we can use to assess vascular reactivity. The technique is called CPT, cold pressor test. Cold pressor test is a test used to assess the body's sympathetic nervous system response. A person's hand or foot is immersed in ice water for a few minutes, which triggers systemic sympathetic response leading to vasoconstriction. Now, you can now measure the resulting changes in blood pressure, heart rate, and forearm blood flow using devices like strain gauge, plateletography, or Doppler ultrasound. Another technique that can be applied is the use of Doppler ultrasound. This non-invasive imaging technique can be used to measure blood flow velocity and vessel diameter in real time, allowing researchers to observe changes in vessel size in response to various stimuli. This is often used in studies of peripheral or coronary circulation to assess vascular reactivity. And we have MRI, magnetic resonance imaging. Magnetic resonance imaging can also be applied to measure changes in blood flow and vessel diameter in response to stimulus. This is a non-invasive method and it can be used also in research to study vascular function in the brain and other organs. Let us now go straight to primary hemostasis. Primary hemostasis typically involve platelet activity, platelet plug formation, and we are going to be looking at different how platelets goes about performing its function during this process of primary hemostasis to form platelet plug. We are going to be looking at platelet adhesion, activation, release of platelet granules, and eventually platelet aggregation. What is primary hemostasis? Primary hemostasis is the stage of hemostasis that prevents blood loss immediately after vascular injury. It involves platelet adhesion, activation, aggregation, resulting in formation of a temporary platelet plug at the site of injury. The initial plug is essential for stemming blood flow and provides the foundation for fibrin stabilized clot of secondary hemostasis. Like I said earlier, platelets is the main thing in primary hemostasis. Therefore, let us have an overview of what platelet is like, platelet structure and biology. Platelets are small anuclear cytoplasmic fragments that board up from megakaryocytes in the bone marrow. They circulate in the blood at concentration between 150 to 450 million per milliliter. The lifespan of platelet, falls between 7 to 10 days. And in intact blood vessel, intact blood vessels with normal endothelium, platelets remain inert. They do not adhere to vessel walls, neither do they aggregate. So, in a normal vessel, blood vessel with normal endothelium, platelets circulate in the within the blood vessels in the blood without adhesion or aggregating. Platelet membrane are rich in phospholipid. On activation, we have phospholipids like phosphatidylserine that flips to the outer membrane, providing a larger surface for coagulation cascade to take place. We have platelet factor 4, a chemokine that is stored in alpha granules and it is released and binds heparin or negatively charged molecules, modulating coagulation and inflammation. Platelet surfaces contain receptors. These receptors are very, very important for this process of primary hemostasis. We have receptors like protease activated receptor 1 and 4. Now, protease activated receptor 1 and 4 found on the surface of platelets are thrombin receptors on platelet membrane. This thrombin cleaves and activates them, triggering strong platelet activation that leads to shape change and also positive feedback that leads to release of more adenosine diphosphate, thromboxane A2, platelet factor 4, and activation of integrin, glycoprotein 2B3A that enhances platelet aggregation. Protease activated receptor 1 mediates rapid high affinity response, while protease activated receptor 4 provides sustained signaling. We have other receptors on the surface of platelet that are involved in platelet adhesion, activation, and aggregation. We have a complex of receptors called glycoprotein 1b, 9, and 5 that binds von Willebrand factor. And we also have an integrin, alpha 2b beta 3 integrin, presented glycoprotein 2b 3a that binds fibrinogen during aggregation and also serves as a receptor for collagen, thrombin, and adenosine diphosphate. Platelet also has granules that play very important role in this process of platelet adhesion, activation, and aggregation. We have alpha granules and dense granules. The alpha granules contain fibrinogen, factor 5, von Willebrand factor, platelet-derived growth factor, and platelet factor 4, while dense granules contain adenosine diphosphate (ADP), adenosine triphosphate, calcium, and serotonin. Platelets are metabolically active despite being anuclear with mitochondria and enzyme system, enabling secretion, signaling, and shape change. Let us now look at process of platelet adhesion. Now, following vasoconstriction, remember that when our vessels, the blood vessel, sustain injury, when there is vascular injury, the first thing that the blood vessel does is to do what? To constrict. The vessel was contract. And like we said earlier, this contraction is mediated by local nerve reflexes and then mediators like thromboxane A2, serotonin, and endothelin. These things are released by the endothelial cells surrounding the vessel wall. Now, following this vasoconstriction, the essence of vasoconstriction is to reduce the flow of blood to the site of injury, thereby minimizing blood loss. Now, following this vasoconstriction, platelets circulating in the blood vessel begin to adhere to the site of injury. Now, the intact inner lining of the blood vessel, that is the endothelium, normally prevent platelet adhesion. But when there is vessel injury, when there is vascular damage, it exposes the underlying subendothelium, the collagen, and the immobilized von Willebrand factor that is present in the subendothelium. Now, platelets adhere to these exposed surfaces through specific receptors, the receptors that we mentioned earlier, found on the surface of the platelet. The number one receptor is a complex, the glycoprotein 1b95. This receptor that is found on the platelet surface binds to von Willebrand factor. It binds to von Willebrand factor, and in doing so, helps the platelet to adhere to the exposed surface of the endothelium. This interaction is particularly important in areas of high blood flow where it helps platelets to roll and slow down. Now we have another platelet receptor, glycoprotein 6, and integrin alpha 2 beta 1. These receptors directly bind to the exposed collagen, providing a strong, stable attachment. So you see, when there is vascular injury after vasoconstriction, because the injury exposes the underlying part of the endothelium, the subendothelium. And when that is done, the von Willebrand factor is exposed, collagen is exposed. This serves as a point of contact for platelet to adhere. And platelets adhere to this particular part of the injured vessel using their receptors. And chief among them is glycoprotein 1b95 complex that adheres to von Willebrand factor at the part of exposed blood vessel, and then glycoprotein 6 and integrin alpha 2 beta 1 receptors that help the platelet to attach to the exposed collagen. Now, the adhesion process is tightly regulated and occurs only at injury sites, preventing inappropriate clotting, in healthy vessels. Once platelets adhere, they become activated. So we are looking at the second process, platelet activation, and then the release of platelet granules. Once platelets adhere, they become activated, which triggers a series of significant changes. One is that once platelets adhere to the point to vulnerable factor that is an exposed vascular site, and the collagen, they become activated. Okay, remember they do this adhesion with the help of receptors found on the surface of the platelet. They become activated, they change shape. Okay, they change from a smooth disc to a spiky amoeboid form with protrusions called pseudopods. This shape change increases their surface area and helps them interact with other platelets. Now, as a result of it, they also in the process activate other receptors that are found on the surface of the platelet. Receptors like glycoprotein 2B, 3A receptors, which are integrins, they become activated and enable fibrinogen binding. Another thing is that phospholipids like phosphatidylserine flips to the outer membrane leaflet, providing a catalytic surface for coagulation cascade. Now, activation also, once platelet is activated, apart from shape change and conformational change and the activation of other receptors like glycoprotein 2B3A that helps in fibrinogen binding, there is also release of platelet granules. Activation of platelet causes release of platelet contents of their granules. And then these granules contain powerful signaling molecules. And some of these granules, the granules that are released when platelets are activated are number one, what we call adenosine diphosphate. Adenosine diphosphate is a potent agonist that activates and recruits additional platelets to the site of injury. We have the second one, thromboxane A2. Now, thromboxane A2 is a lipid compound that further promotes platelet aggregation and vasoconstriction, creating a positive feedback loop that amplifies the hemostatic response. We have another one, serotonin. Serotonin is a vasoconstrictor that reinforces the initial vascular spasm. So we have adenosine diphosphate being released as part of platelet activation, thromboxane A2 being released, serotonin being released, and then calcium is also released that helps in secondary hemostasis. When these content of the granules are released, remember part of their function is to recruit more platelets to the site of injury and also enhances the process of vasoconstriction. So the final stage of primary hemostasis is the formation of weak plug. Okay, the activation of platelets leads to a conformational change in the alpha 2b beta 3 integrin, which is specifically glycoprotein 2b 3a receptor, making it active and able to bind to ligands. Now, the most crucial one thing I want you to note at this point, the most crucial ligand for aggregation is actually fibrinogen. Now, what does this fibrinogen do? Fibrinogen acts as a molecular bridge linking the alpha 2b beta 3 receptors like glycoprotein 2b3a receptor of adjacent platelets together. When it links them together, they come together to form a mesh-like structure. This mesh-like structure is actually what goes on to become what we know as platelet plug. This process of platelet binding to each other forms a temporary and fragile platelet plug that provides a temporary seal to the site of injury. Now, positive feedback from adenosine diphosphate, thromboxane A2, and thrombin further amplifies recruitment, leading to rapid plug growth. This plug is then stabilized by the coagulation cascade during secondary hemostasis to form a stronger, more stable clot. Okay, so we have it now that when there is vascular injury, there's vasoconstriction, and this vasoconstriction is mediated by the local reflexes and then thrombosin A2 serotonin endothelium produced by the surrounding endothelial cells. And immediately after that, platelets are attracted to the site of injury because the endothelium is now exposed, exposing the subendothelium that contain von Willebrand factor and collagen. The platelets, with the help of their receptor, precisely glycoprotein 1b complex receptor, binds to this von Willebrand factor, and then glycoprotein 6 also binds to the collagen, causing adhesion of platelet to the site of injury. Once platelets adhere to the site of injury, they are activated. Now they are activated and they undergo conformational change. They change shape, providing larger surface area for more reaction to take place, and also activate more receptors on the surface of the platelet. These receptors integrin like glycoprotein 2b 3a that helps in linking platelets together, and is able to do that by the help of fibrinogen. And then you have, once platelet is activated, they also release their content, the content of their granules, which are adenosine diphosphate, thrombosin A2, serotonin, even calcium. These molecules, they are vasoconstrictor, and they are also help to promote or enhance platelet aggregation by recruiting more platelets to the site or attracting more platelets to the site of injury. Now, once this is done, the receptors glycoprotein 2b and 3a helps to link the adjacent platelet together with the help of fibrinogen to form a mesh-like structure, what we call what? Platelet plug. This platelet plug is not that strong. It is stabilized by the secondary hemostasis, which we are going to look at a later date. Okay. Now let us look at regulation of primary hemostasis. How do we regulate this process so that they don't escalate and cause more injury or do more than they are expected to do? Now, regulation involves a critical balance between promoting platelet plug formation at the site of injury and preventing its uncontrolled spread throughout the vascular system. This is mainly achieved by the healthy entered endothelium of blood vessels, which actively secrete substances that inhibit platelet activity. Platelet inhibitors, we have things that actually inhibit platelet activity, and these substances are produced by the endothelium. The chief among them are nitric oxide and prostacyclin 2. Now, prostacyclin 2 is a powerful vasodilator and the inhibitor of platelet aggregation. It works by increasing cyclic adenosine monophosphate in the platelet, which, like nitric oxide, suppresses platelet activation. So prostacyclin 2 suppresses platelet, inhibits platelet aggregation and suppresses platelet aggregation. And it is a vasodilator, and it helps in regulating platelet actions during primary hemostasis, preventing the process from escalating. We have another platelet inhibitor and a potent vasodilator, which is nitric oxide, and exhibits its function by stimulating guanylate cyclase in platelets, leading to increased level of cyclic guanosine monophosphate, which reduces intracellular calcium levels and inhibits platelet activation. So nitric oxide also inhibits platelet activation and also is a vasodilator. And it does that by increasing the content, the cyclic guanosine monophosphate content of the cell. And when the level of cyclic guanosine monophosphate within the platelet is increased, it acts as a powerful inhibitor, leading to decreased intracellular calcium inhibition of platelet activation, and also causing vasodilation. We also have feedback mechanism that helps in regulating platelet function. The hemostatic process also contains internal feedback loops that both amplify and regulate the response. While initial platelet adhesion and activation lead to release of pro-aggregatory factors like ADP and thromboxane A2 in a positive feedback loop. This is localized to the site of injury by these inhibitors. The circulating inhibitors from the healthy endothelium prevent the spread of this activation beyond the damaged area. We also have role of some of the platelet receptors in regulation of platelet activity. Okay, resting platelets maintain an inactive form of glycoprotein 2b3a receptor, that is integrin alpha 2b alpha 2b beta 3 receptors, preventing them from binding to the fibrinogen and causing platelet aggregation, but only upon activation at the site of injury do these receptors undergo the conformational change needed for aggregation. Now we have natural anticoagulants such as antithrombin and protein C system that act in parallel to prevent uncontrolled clot propagation. Let us now look at laboratory tests that we can use to evaluate primary hemostasis. We have different tests that we can use to evaluate primary hemostasis. Number one, we have what we call platelet count. Platelet count is a routine test that assesses the number of platelets in circulation, the quantity of platelets in circulation. And the low platelet count, what we call thrombocytopenia, can lead to bleeding disorders. We have another test that assesses platelet function. The platelet function like another test like assessing platelet function like bleeding time test. This test measures the time it takes for bleeding to stop after a small puncture in the skin is made. It reflects platelet function and vessel integrity. However, this test is no longer being used frequently as today because of its variability. We have another test that assesses platelet function using what we call platelet function analyzer, PFA 100 or 200. This particular test is more sensitive than bleeding time test, and it measures the ability of platelets to form plug by stimulating conditions of blood flow and collagen exposure. It is an automated test that provides a comprehensive assessment of platelet function, and it can be used to diagnose some disorders, platelet function disorders, and von Willebrand disease. Now we have another test that we can use to assess platelet function, like platelet aggregation studies using platelet aggregometry. This is the key test that directly measures the ability of platelets to clump together, that is, ability of platelets to aggregate in response to various activating agents like agonists, like ADP, adenosine diphosphate, collagen, and epinephrine. It is considered the gold standard for diagnosing many inherited platelet disorders such as Glanzmann thrombocythemia. Now we have another test that we can also use to assess not platelet function directly, but ability of platelet to adhere, because von Willebrand factor is needed for platelet adhesion. If it is not present, platelet will not be able to adhere to the site of injury. So, since von Willebrand factor is crucial for platelet adhesion, tests that measures its level and function are essential for evaluating primary hemostasis. So we can assess von Willebrand factor antigen, which measures the total amount of von Willebrand factor protein present in the blood, or we can also measure its activity that measures how well the von Willebrand factor can bind to platelet receptor glycoprotein 1b. And abnormalities in this test can indicate von Willebrand disease. This primary hemostasis If there is any defect in platelets or their receptors, there could be bleeding disorders, which often presents as mucocutaneous bleeding like petechiae, epistaxis, gum bleeding, menorrhagia. We have key disorders that can occur as a result of platelet dysfunction, like thrombocytopenia, when you have a reduction in the amount of circulating platelets, or vulnerable disease, Banner-Stollier syndrome, Glanzmann thrombocythemia. We are going to look at these various disorders in detail in future lecture. I believe that you have been able to, you can at this point, you can actually say the processes that are involved in primary hemostasis, the vasoconstriction, and then the platelet function with regards to primary hemostasis. When I mean the platelet function, I mean the platelet adhesion, activation, and aggregation, and then ways that these processes can be regulated. And of course, you can mention different laboratory tests that can be used to assess this particular process, that is primary hemostasis. We will be discussing secondary and tertiary hemostasis in the next lecture. Please keep a date with us. Don't miss that particular lecture. It is a continuation of what we are discussing today. Remember, the topic is hemostasis. Thank you for joining us. And if you have not subscribed yet, please, this is time to subscribe. And if you have questions and feedback, please feel free to drop it on the comment section and we will speedily get back to you. Thank you very much. These are my references. Thank you all. Thank you.